morning, everyone. My name is José Mauricio Fernandes. I will be sharing this session, and Diego is helping me there. I just want to remind you that the presenters that they have about 10 minutes, okay, for the presentation, 2 minutes for the discussion, and we will have about 7 people today here. 4 are present, and 3 are virtual. Okay, so we're going to start.
Okay. Hello, everyone. I'm Wei, and I'm from the Humboldt Center for Environment Research in Germany. And it's my honor to give this presentation to share my ongoing work. Today, my topic is about the attribution of the 2022 European maize failures to the anthropogenic climate change. Okay. As we all know, in recent years, there are more and more extreme climate events. Like in 2022, Europe have experienced one of its worst drought heatwave events. Like in 2022 summer, there is a very low soil moisture, a low VPD, and a higher temperature. This kind of extreme events have induced the have induced the maize yield loss. According to the FAO data, the maize yield in 2022 is the lowest in the last 5 years. So we can think it's a crop failure event. And recently there is a new paper. It's have found that the anthropogenic climate change have contributed to the drought contribute to more than 30% of the drought intensity and the spatial extent of this drought heatwave event. So we want, however, we want to know whether and how much the anthropogenic climate change contributes to the yield loss, especially for the crop failure events in 2022. By determining such kind of contribution, we can maybe we can provide an valuable insight to adaptive planning for such similar events in the future. Actually, the attribution analysis have developed rapidly in recent decades, like the climate attribution work. They try to address such questions about what's the contribution of anthropogenic climate change to the observed trend or the magnitude, or the probability change in the weather or climate event. And in recent years, the research have gone a step further by linking the observed impacts in the human management system to the observed climate change. So here, and it's called the impact attribution. So here we follow the impact attribution framework. We want to try to attribute the crop yield change to the anthropogenic climate forces. Here is the general framework of the impact event attribution. First, we need use the impact model to get the factor simulation. Here shows the solid blue line, and then we need to compare the factor simulation with the observation to make sure that the factor simulation is reliable. And then we can use the counterfactual climate data to force the model get the counterfactual simulation. Here, the counterfactual climate is represent that the climate conditions without the human influence. Then we can compare the factual simulation with the counterfactual simulation. The difference between these two, we can call the contribution of the human influence. However, there are also arise two challenges. The first one is about the construction of the reliable counterfactual climate forcing, and the second one is about the reliable factorial simulation. Because the attribution analysis is relies on the high explanatory power of the impact model. So to deal with the two challenges, first, we use the dirt change method to create the counterfactual climate forces. Because the time limit, I want to just show the general idea of this method. Because the observed trend in climate is a combination of the human-induced trend and the and a relatively trend in natural variability. So we consider the ensemble mean of CMIP6 as the human-induced trend, and then remove the trend from the observed time series. Then we can get the counterfactual climate forcing to force the well-calibrated crop model. Then we can get the simulation to do the further analysis. As for the impact model, we choose the organic crop model. This model is well calibrated and evaluated in Europe, but in this work, we still do the calibration work to make sure that the model can capture the yield variability, especially the yield loss. And we also calculated the metrics like the correlation coefficient and the root mean square error for the observed and the simulated time series to find the optimum parameter combinations. So here is the whole workflow of my research. Next, I'll show the preliminary results of the first part. First, we use our data to identify the climate condition during the 2022 maize growing season. We found that during the maize growing season, there is a higher mean temperature and maximum temperature in the whole Europe, and also there is a lower precipitation, lower soil moisture in the western and the southeastern Europe. So we think this hot dry condition is the main driver of the yield loss in 2022. Next, about the model performance, we choose the optimum parameters with the maximum correlation coefficient and the minimum RMSE. We can see from the picture that after the calibration, the model can the correlation between the observed and the simulated time series have increased, and also the model now can capture the yield loss in 2022. We also compared the results from different trending methods, like the linear detrending, the lowest detrending, and the moving average detrending, and there is no significant difference between the different methods. We also analyzed the simulation results for the top six maize producing countries in Europe. We can say that after the calibration, the model now, for the most countries, the model now can capture 10 to 50% of the yield variability. And we also calculate the metrics for each rate. After the calibration, the correlations between the observation and simulations for each grade have increased. And for the RMC, even though the mean of RMC have no significant changes, but we can see the distribution, it have a shift to the left, which means that the RMC has decreased. So in summary, after the calibration, the model now can capture the yield variability and yield loss, not only in the regional scale, but also for the country scale and the grade scale. Then we use the calibrated model to evaluate the 2022 yield anomaly. Here is the spatial pattern. We can see that in the Western and the Southern Europe, the maize anomaly in 2022 is lower than the year before. Here is the 1990 to 2021. However, we can notice that the spatial pattern from the lowest trending is very different with these two others, maybe because the choice of the window size, or maybe because this method is not proper for our research or data. I need to further check. Here is the work that already done, and the next step is to following the dirt change method to create the counterfactual climate forcing, and then force the model to get the impact data, and then we can analyze the data and size, like the magnitude, the spatial extent of the crop failures, and analyze the variations, and maybe do further sensitivity and analysis. Okay, thank you for listening. And because this work is at very beginning stage, and there many details need to be improved. So any discussion and suggestion are welcome. Thank you.
Time for questions.
I know there's many different ways to construct counterfactuals. Why did you decide to go for the detrending method and not just use natural forcings only climate data set?
Because well, I choose the dirt change method because as I say that we consider that the observed trend is not only about the human-induced trend, but also the natural variability trend. We can, through this method, we can separate the human-induced and the natural variability. And I also know that in the AgMIP10 there is a, I don't know how to pronounce it, the HPC method.
No.
Well, yeah. Actually, that method is also very good, but I think it can't isolate the trend from the natural variability. But in my design, I also in the further analysis, we also try to use the AgMIP10 data to force the model and then compare the difference to see whether there is a big difference.
The AgMIP data set also has pre-industrial climate. So you could also use this one as a third comparison point.
Yeah, I also want to use this one.
So that's fine. I think we move to the next test.
Okay, thank you. Again?
Yeah.
Please.
Yeah, thanks so much. My name is Fikran Mariam. I'm a postdoc at TSRO, Australia, based in Canberra. So the title of my talk is Productivity and Profitability of Legume-Based Cropping Systems in Southeastern Australia. Yeah, the reason why we are interested in this work is because we believe that integration of legumes in the cropping system is not enough or low in Australia or elsewhere. There are many reasons for that, but among the major ones, there is really very limited data on their long-term benefit in terms of productivity, profitability, and ecological aspects. And this is where modeling can offer a good job and predicting the long-term benefit of these systems. Then another one is market uncertainties for legume crops, and they're also relatively susceptible to disease and pests. And also climate variability is one of the challenges. Yeah, and we know that legumes have multi-advantages or benefits to the system, from increasing productivity, enhancing sustainability, and so. Just to give an example, in Australia, growing pea and lupin. Sorry, so pea and lupin in the system can contribute up to 100 kgN to the next crop, and growing legumes in the system can also reduce the pests and the disease can happen in the coming growing season. Yeah, so the objective of this work is to provide a long-term evidence on legume-based cropping systems in terms of their yields, soil attributes, and profit. And this work is actually based on a long-term experiment at one of our CSRO sites in New South Wales, which we evaluate 4 different systems of crop rotation. So I took data from this one to calibrate AgMIP10 and provide a long-term prediction. So for this work, we have 4 different scenarios. The first one is the canola wheat system, which is a conventional system. And we have 2 other different options where fabin. Sorry about this. We have 2 different crop rotation systems where fabin and lupin considered as a pulse here. We have another one, the third one, with fabin only as a pulse crop. And the last one is a lucerne-based crop rotation system, because option 2 and 3 are intermediate between the 2. I would only focus on option 1 and 4, combined with different end targets during tree flowering and across a long-term climate, and also across a contrasting size in terms of rainfall. For this talk, I only focus on 2 contrasting sites, Borowa and Condoblin. They are contrasting in terms of their rainfall. And then I use data from the field to set up AgMIP10 and calibrate and evaluate so that we can have a prediction on the long-term yield, profits, and soil organic carbon for all the systems. Yeah, so we have a lot of results, but for this talk, I will only focus on the yield, the soil organic carbon, and the profit. When we see the yield result, as you can see here, the red one is the one with lucerne, and the blue one is the canola wheat system, the conventional one. As you can see here, the yield is consistently higher for the lucerne-based system, especially below 100 kgN input. But after 100 N, then the difference becomes small, but even higher for the conventional system. Wheat 1 is the first wheat in the crop rotation system, and wheat 2 is the second wheat in the crop rotation system. So this one is under low rainfall condition. At high rainfall condition, again, the lucerne system is consistently higher, except at high N, which is 300. And here, which is at high rainfall, then, and also at wheat 2, the lucerne system is consistently higher all over all N levels. And when you see the response is better for Borowa, which is high rainfall, than Condoblin. This is because of the good soil moisture. And again, the response was better for wheat 2 than wheat 1, because there is a better N legacy for wheat 1 than wheat 2. So the response is better for wheat 2. And when we see the soil organic carbon, the lucerne-based system can substantially build the soil organic carbon compared to the conventional one, regardless of the end rate, but the response or the difference becomes small after 100 N target N. However, as you can see here, even the conventional system can drain or mine the system under 0 input or no input.
quick question in between. Yeah, please. Is it simulated or is it observed now?
This is a simulated one.
Okay.
Sorry. Yeah, so as I said, the first focus was for 4 scenarios, but for this talk, we only took 2 of them. So I did a profitability analysis for 2 of them. So as you can see here, at low rainfall area, adding nitrogen was not totally profitable, even declines for condobolin, and it stays stable here and becomes decline for Borowa. So adding nitrogen for the Lucerne system was totally not profitable. However, increasing N was profitable until 100 N and becomes decline, and it keeps increasing for the Borowa, the high rainfall area, because of better yield. And in this case, adding nitrogen costs us a lot, so the profitability was lower. So when we see the system overall, especially at low input, the Lucerne-based crop rotation was better compared to the conventional one. Yeah, so integrating legumes into the system was profitable, and we can increase productivity by improving soil organic carbon, especially at lower inputs. That's the conclusion we have.
We have time for questions.
Quick question on the last slide before this one. The effect on the gross margin, is this for the entire cropping system or just the wheat part?
The whole system.
The whole system. Yes.
So I'm really impressed by the buildup of soil organic carbon achieved in the simulations. If I think of observed field trials, I've rarely or ever seen any agricultural system that increases soil organic carbon over time. Do you know what the observed values would be for these locations?
Yeah, as I said, we calibrated the model based on the field observation, but that was for the data for one year. Yeah, but because, as you know, Lucerne has a higher biomass and it can fix quite a lot of nitrogen, so the soil organic carbon keeps increasing as long as there is no limitation due to rainfall. Then it keeps increasing.
Saturates.
Maybe there's more shots. That more questions. Yeah, that's. It's easy. The other question. We still bit of time.
Thank you.
Thank you.
Henrik?
Yep, that's me.
Don't talk to him.
That's what I said. So
Going in PDF, that was the other PowerPoint. Can you find to the edition? Or find my own presentation? It's on the Google Drive. I think it's already open. So some PDF as well. That's me. Yeah, yes. The presentation mode to go see the share screen. And I want first. So, yeah, that's tough. Okay, perfect. Yeah, my name is Lennart Jansen. I'm a PhD student at the PIC and the University of Kassel, and I want to present some preliminary results on attributable and projected economic losses for German agricultural production. As you can see, it contains the word attributable, so attribution. I'm really happy that the first presenter already gave a nice intro on what attribution and impact attribution is. So I won't go into too much detail here. Typical impact attribution approaches in agricultural science focus on yields, which are related to changes in weather, and those are due to anthropogenic climate forcing if you set up the factual and counterfactual climates in the way that either she did, or you can use climate data sets from models that have been forced without the anthropogenic emissions part. So in theory, this gives you a nice, short, clear impact chain, and yield changes are easy to interpret, but they are also an incomplete picture of the impact on an agricultural system. So I wanted to focus on changes in agricultural production and resulting economic losses and gains, but this makes the impact chain, of course, longer, more complex, and it can bias your results to a great degree because yields are area independent, production is not. So yield changes could be large, but if it's a niche crop only cropped in small areas, the final impact will be small. But you have to include more of the agricultural system. That is good, in my opinion. You have a better reflection of climate change impacts on food security, on agriculture or the wider economy, as well as livelihoods of farmers. Problem is, agricultural production is not just yield times area. It depends a lot on the management and adaptation. A farmer will not crop what isn't profitable and what isn't suitable for the local climate. And farmers have lots of experience, so they know this. This makes the design of the counterfactual quite complicated. Typically, as climate scientists, we take the academic approach. We keep it really straight. Everything is the same except the climate. So we isolate the climate signal. But this is very academic view of the world, does not reflect reality. This ties also back into the next simulation round of the GGCMI team, which want to include adaptation in their climate change impact studies into the future. And I didn't plan for this, but it's a nice link now that I was at AgMIP. And I want to do this for the counterfactual. So I want that the farmers can decide according to profit and the gross margins that what they plant, not only in the factual or the reality, which they are doing, but also in the counterfactual scenario. Now, crop model can't give me this information. It will only output yield. So I need a second model, a land use model or an econometric model. And that's what I collaborated with colleagues from the University of Hohenheim. They have a field-specific optimization model. This resulted in this huge, complex modeling chain. Well, it's not that complex, but I show every input data that I use. So the crop model I use is AgMIP10 or NextGen. It's forced with climate data from both factual and counterfactual data sets from the AgMIP data sets. Management data is kept factual. Well, for the counterfactual scenario, it's a bit of a mix because we adapted phenology or sowing data a little bit, but differences are really minor. And soil data we get static because the link to climate is really long-term and not really relevant for timescales that we are dealing with. And the crop model then outputs yields for both scenarios. And the field-specific optimization model takes these yields, the available overall crop land for each spatial unit. In this case, this is this grid, and the input costs to calculate the gross margin for each crop everywhere, and then it maximizes the net present value of all crops in this cropping area. And that is then how it allocates which crop is grown where or what the area shares are. And it has been validated for market-oriented agriculture in Germany because there the gross margin is very strong determinant of what is cropped where. And that is the aim now to do this also for the counterfactual climate. So you have a side result of if there really is significant difference in the cropping areas of the different crops, etc. Of course, you need to provide a lot of optimization constraints here. Not every crop rotation everywhere makes sense. Potatoes are quite grown not in every area. Maize will not be grown in high altitudes in Germany. And it reserves a share of each cropland area for livestock feed demand. Yeah, and in the end, with the areas, the yields, you can calculate total production. Using factual market prices, you get an economic value, and then in the end, attributable economic losses and gains. Now, it would be great to have counterfactual market prices, but that ties into really macroeconomic considerations of fertilizer prices, etc. So that was, for me, definitely out of scope, but maybe I talk to Hermann and we make something happen in the future. And yeah, with that, I can show you preliminary results of the calibration. Because if you want to assess agricultural production or even total agricultural production, it's not enough to focus on one crop. You need to cover a whole range. This subset, you see grain maize, silage maize, potato, spring and winter barley, because we wanted a summer cereal to have the option, the crop rotations, winter rape and winter wheat, cover more than 70% of agricultural production output in Germany. The other big share would be grassland, but that's not part of crop rotations. And sugar beet. Sugar beet doesn't have the model in AgMIP10, so we couldn't include it. And even calibrating 6 to 8 crops is already so much work. I really underestimated this. And you see already a problem here, especially with silage maize and potato. It's systematically underestimated. Getting the model up to 60 tons of yields has proven quite challenging. And this is problematic for the econometric allocation of cropping shares, because silage maize is a really big part of agriculture in Germany. I think it's 40% of total production. And if you systematically underestimate its yields, the econometric model will, of course, not work as it should. So I can't, unfortunately, present to you the economic losses today. I could, but they wouldn't be meaningless in my opinion. But what I can do is show you the real production impacts between the two scenarios. So this is a closer look at all the other crops. You see there's over and underestimation everywhere. Sometimes we capture the trends really well, sometimes we do not. So what I did to mitigate this a little bit is I averaged for a period of 20 years. And this looks already much better, especially if you look at this as a map. And here we have the simulated total production output. So I summed up all the crop yields with the factual areas for which we have data and compared this to the observed total production output. And you see that we capture the spatial yields quite well, even though the simulated production is lower than the observed, which makes sense because we underestimate silage maize and potatoes, which are popular crops in the north. Those silage maize mostly is grown everywhere. And you can also see that the smaller the districts, the lower the output is, which shows the bias that area introduces if you look at production, because they have less area. So of course, production will be lower. And now we can look at the difference to the counterfactual, the climate without anthropogenic forcing. And you don't see a lot of difference. This is mostly due to this bad logarithmic color binning I've done. So I created a map with continuous colors. And there we see spatially heterogeneous impacts already. So the north, which is highly productive, has negative impacts. And the rest of Germany has low to even positive impacts of climate change. Now, it is going to be really interesting to try and differentiate why, because this is made up of multiple crops. So which crops are the drivers, which of the gains and the losses, and how is it related to area? And finally, what does the econometric model contribute in the end if we redistribute the area shares too? So you can force the AgMIP model as well, of course, with projected climate data. So this is SSP3, decades 2050 to 2070. And you see there's no positive impacts at all anymore. There's even really, really negative impacts. But static management, as I said, can really assume that farmer grows the same things in 50 years that he does now, or she. So these are quite uncertain, but give a little bit of a distressing picture into the future with current management. So adaptation, I think, is paramount. And with that, I come to a close. My conclusions. My aim was to assess economical production impacts of climate change in Germany. That's still my aim. My preliminary results show that we can capture historical spatial trends of yields well for 6 crops, but 2 important ones not. So that's a hurdle I have to fix and overcome. Simulations suggest current climate change impacts are spatially differentiated. Projected impacts are more pronounced and negative, but uncertain. And for me, the next steps would be to improve calibration, quantify the uncertainty, and finally derive the attributable economic impacts using this field-specific optimization model. Thank you.
Thank you.
Do we have time for questions?
Yeah.
Thanks very much for your also for your brave attempt to solve different crops. It's still a selection only.
Yeah.
If you go to that detail, I wonder whether you also thought about considering the range of varieties that we have within a crop.
Yes.
If you look at wheat, we have more than 100 varieties registered in Germany, and if you look at the range of phenological responses, there is quite a difference. And there's also an adaptation going on. Farmers already adapt by changing, by using early or late varieties to ongoing climate change. So is that static in your approach?
Very good point. Was no time to
Maybe I have another one. Yeah.
really go into the calibration. What we did is we used the soil climate zone delineations, the Boden-Klimaräume, and fit cultivar parameters for each soil climate zone for each crop. We tried also to fit them by decade to simulate adaptation or evolving variety breeding. That didn't make a difference though in the calibration results. But yes, so I didn't show a map, but the soil climate zones are really complicated and disjointed, but they
Within one zone, you have one parameter set for
one crop. Yeah.
On the distribution of maturity classes.
No, sadly not. There's effort to simulate all of them would be incredible. I think even now it takes a long time to run.
Can I have another question?
It's John.
Yes, short one. When you showed the production results, I didn't quite understand it. Is it a summing up? Is it a metric across the different crops that you are looking at?
So what we do is we average aggregate the yields using 2 districts. Because for district we have
crops.
No, for each crop.
For each crop.
And for each crop we have a data set that has the crop area per district as well as the Duden et al. District. A data set for 30 years. And then we average that over 20 years, and then you get the production for each crop, and then we sum that to get the total production for that district.
But is there not a methodological issue if you bring the sum up or average the production of the different crops, because they are relatively different? There are quite some distinctions between like the yield of rapeseed and the yield of potato in terms of the tons. And so you may introduce an artifact in your results by just bringing those together and don't sort of I missed the term here. So accounting for the differences in the productivity of the different crops that they naturally have.
Well, that of course disappears if you aggregate it. So winter rape has really low yield compared to silage maize. But I think if you want to look at production or total production, that is unavoidable in a way, except if you disaggregate this again by crop.
We can normalize it, perhaps.
Yes.
That was the word I was using.
Okay. Yeah, we can talk about this later. Sure.
Yeah. Have to move on.
Sorry.
Next, Professor Pavin. Here.
Morning, can you hear me?
Yes.
Okay, Maurice, can you stop sharing so I can share my screen?
Yeah. Sorry. Stop sharing. Stop sharing. Now you can share your screen.
Okay. Let's see here. I hope you all can see my screen now.
Yeah, you can.
Okay. So, good morning, everyone. My name is Willington Pavan. I'm assistant professor in crop modeling in CIS University of Florida. I'm going to presenting today simulating Fusarium head blight risk and mycotoxin contaminations using integrated DSAT GDM as generic disease module framework. So before I start, I would like to just start with this slide that is well known for everybody, that we have increasing population And all predictions and all scenarios that we have a reduction in production. And one of the basic factor for that is because of diseases that is cause losses on production. So based on that, I put here this slide just as a discussion that is well known. The people, a lot of people say that all the modules are wrong, but some are useful. And that's true when you are in real life. For example, we have hurricanes here in Florida, and all the modules gave us different directions, but at the end we can use those results to make a decision and to mitigate any problem. So they are useful. So based on that, came to the crop modeling and disease modeling. Build on these. This is a lot of crop modules. They fail to account the biotic stress, so which can result in inaccurate predictions. So my work and my program and the team that we work together, we are looking for to mitigate this effect of biotic stress. So is well known that the wheat production losses due to the pests and disease damage estimated from 21 to 31% in wheat. And climate change is likely to exacerbate the biotic impacts in the crop production. So incorporation of these modules, disease modules, and crop modules, integrate them is something that's needed. So this is what we as a group are developing in our project. So a few years ago, is about 20 years ago, if I can say, we started to develop a generic disease module that was, the main idea was to simulate different diseases and multiple diseases across different crops. So we implement that in 2000, from 2004 to 2007. So we developed that and keep improving it. So considering many factors like temperature, relative humidity, and inoculant presence, and also and we simulate the cycle of pathogens, including spore production, dispersal of these spores, and infection and disease development stage. So our idea was to develop something that could be reused for different diseases. So we allow parameterization using a file where we can provide all the information about the disease and parameterize for each individual case. So, and we start to, is well known that DSAT decision support system as agricultural transfer is a suite of crop modules that mimics in a process-based module, mimics the development of the plant. So based on that, the success of the DSAT, we started to think to integrate our disease module with the past subroutines that we have inside of the DSAT. So we started developing and using the past coupling points that we call in the DSAT. It was developed, start thinking in 1993 by Bachelor and the UF team, that they started implementing these coupling points inside of the DSAT. So many crop modules, like the family from crop grow modules, they have already implemented all these coupling points, but others, like for example, the series family and other crop modelings, they don't. So we started developing these coupling points inside of some main crop modules like in wheat that we implement. So the coupling points following the same idea that was proposed by Bachelor et al. And in 1993. So in 2021, we published these modification implementation of the coupling points in wheat. So we already finished the coupling points for crops in wheat, cereals wheat, and cereals rice. So those coupling points allow us to affect the plant in a secure way that we are not mess up the module. So we provide information for the crop module related to each coupling point. So based on that, we decide to translate the generic disease module that was written in Java originally to C++, allowing us to connect DSAT and the generic disease module in a certain way that we could have a monolithic approach that we can run and use the speed of the DSAT to have the coupling the disease modules and get the results. So our idea was to implement in a certain way that we can replace the modules and run again. So here is some examples of the inputs and the configurations that we do in the DSAT. So how it works is we have the DSAT and we have the generic disease module that in a daily basis communicate with the DSAT, getting the information that came from the past subroutine and process the information and using the weather from the DSAT automatically and send back the information to the plant. So one of the case study that we did, using this approach was published last year in Nature Climate Change, where we have the publication here, as you can see. Okay, you can access later. But based on this, I'm sorry. I'm not able to change the slide. Okay. So based on going further than that, we decided we need to improve because we need to run different diseases at the same time. So we need to improve and expand. So we start to these improvements is thinking in epitype, that is a type of disease that we have. So we started having, like, for example, for leaf and stem, we have the leaf stem and yellow rust, for example, with blast and fusarium head blight. So our idea here at University of Florida is to use leverage the DSAT as with new coupling points integrated with the generic disease module, implementing new diseases and running parallel those diseases and affect the module, and run a partnership with some partners that has data available, so we can run different scenarios in different places around the world. And we are leveraging the DSAT PyTia, and Hypergear, our supercomputer, so we can run those modules on the internet. So we started doing fusarium head blight study and implementation. So this module, fusarium head blight is a huge issue that is attacked the wheat and all around the world and produce a toxin, deoxynivalenol, DON, is known as DON, and attack the plant and produce toxins that is not good for human. So we started developing that. So in 2005, as you can see, we already had a module that was developed by Del Ponte Emerson and Mauricio Fernandez and myself, and we developed that module in 2005, and that give us an idea about the production of spores, the cloud that we call about the spores. But that is used relatively immediate. That's a very very expensive information. So we started making correlations between the soil moisture and the pressure of the disease. So based on that, we are started using information from the DSAT, where we can have the moisture and the crop residue and create the spores in the cloud. So we run an experiment about using a Turuk cultivar that is used in Brazil, in South Brazil, integrated with all the data that we have, and we generate for a region in South Brazil that is Planalto Médio, and we implement those simulations. And you use the GSAT is a web tool that we can use to generate and collect the data automatically from NASA Power, CHIRPS, and Soil Global Database, and we run simulations. So we were able to simulate the presence of fusarium head blight in many places around around the region, the Planalto Médio. So we could be able to generate outputs, and so that outputs, and you see red is the normal crop module, and in green when you have the disease running together. So we could simulate and mimic the results. Our next ongoing work is to, as we have the mass of the disease running in the system, so we are correlating that information with the results to get the information about the toxins that's produced by the modules. So that's my presentation for today. I'm open for questions, and also open to be contacted later to work together.
Questions? Any question?
Yes. I thought that was amazing. Crop models definitely need more ways to deal with disease, which is a very pressing problem for farmers. Have you any plans to make this work expand to other diseases as well?
Yes, we have a plan to expand to other diseases because, as I said, genetic disease module, it's parameterized, so you can parameterize for others. For example, we made the studies for potato, we have for wheat, we are thinking on rice, so many diseases in different crops and different diseases with a different cyclists we call, like different types, a monocycle, polycycle, and if you have, for example, one disease, one place that you generate the spores and it's transported by the air to another place, so we are also able to run that at the same time and make it the simulation.
What time we stop?
Apologies if I missed this. Do you have a resistance parameter for the cultivars you use, or do we need to introduce one now?
Yes, we have parameters for resistance, and our idea is to change a little bit on the DSAT parameters to allow us to make it to tell if that variety is resistant to the disease or not. So you can have a different results as based on the inputs. Nowadays, we made all the informations on the configuration of the system, so you can tell that about the resistance, host factor resistance.
Amazing, thank you.
Thank you.
Thank you.
Thank you. I'm going to stop sharing here.
Okay, thank you. Okay, let's move. Jose?
Yeah, hi everyone. I'm gonna share my screen now.
Okay.
Okay, are you able to see the presentation?
Yes.
Okay, fantastic. Great. Well, I hope you all are having a good afternoon. So my name is Jose Guarin. I'm a research scientist at the NASA Goddard Institute for Space Studies. And today I'm excited to present to you some of the work that AgMIP Ozone's been doing on simulating the impacts of tropospheric ozone and climate change on global agricultural production. So with that being said, most of this is going to focus mainly on the AgMIP ozone phase 2 work. And that's going to be evaluating the climate change impacts on current and future global agricultural production using global gridded crop models that have the ability to simulate ozone stress. So I'm sure you're all familiar with crop models, but not all of them can simulate ozone stress. We would like to change that here in AgMIP ozone. And so the approach is based on AgMIP GGCMI Phase 3, the climate change assessment led by Jonas Jägermeier that was published in Nature Food 2021. So we follow that framework. And then we also build upon the AgMIP Ozone Phase 1 activity, which is being led by Lisa Emberson at the University of York. And that's essentially looking at a multi-model ensemble of wheat crop models to evaluate if they're able to reproduce the ozone responses that we're seeing in field experiments across the globe. And so building upon those two, our goals are to provide the decision support for future global food production and ozone mitigation strategies, and also raise awareness of the global ozone impacts in the agricultural community, because depending on where you are, if you're in a high ozone concentration region, there could be significant impacts. And just to give an idea of the participating teams and the interest, so we have 13 teams that are interested in participating and 5 that are already participating that have started the simulations. So that's fantastic. It's great to have this kind of ability to collaborate across modeling teams with AgMIP. And if you're interested, I'll have some details later at the end to join if you have a global grid crop model that you would like to participate in this study with. And so just a brief review of the protocol. The full protocol is available online at the GGCMI Redmine. So the full details can be seen there, but just a summary. So we're simulating a historical and 2 future shared socioeconomic pathway scenarios with an ozone effect. And we're looking at an ozone sensitive and an ozone tolerant cultivar classification. So I'm not going to go into all the calibration details here because I don't have time, but at AgMIP Ozone, we have field experiment data and literature data for modelers to calibrate their models to develop ozone parameterizations for an ozone tolerant, an ozone intermediate, and an ozone sensitive cultivar classification. And so here we're looking at the tolerant and the sensitive classifications to get the range of the global ozone impact. Ideally, we would like to have a map that has like all of these different areas like the typical cultivars, like whether or not they're ozone sensitive or tolerant, but that doesn't exist yet. So huge area for exploration there. But for right now, we're just going to look at the range. And so this protocol consists of 5 scenarios. So we're going to be doing a historical with dynamic ozone effect, which is going to be our baseline, and that's going to be our calibration scenario. Then for the second one, we asked modelers to do a historical, the same exact scenario, but without ozone effect. So turn it off like in the model if they have a switch, or just remove the ozone column, however they want to do it. But this will be a proxy for pre-industrial ozone so that when we compare scenarios 1 and 2, we can see the ozone impact during this historical period. Then we move into the future scenarios. And so number 3 is going into the future, which is 2015 to 2100, using dynamic ozone for the low emission scenario, which we consider SSP 1 2.6, which we consider the low emissions, and SSP 3 7.0, which we consider the high emission scenario. And so this scenario is both the combined future ozone and climate change impact. Then in order to isolate ozone impacts, we do scenario 4, which is the future but constant 2015 ozone. So when we compare the two, we can see the ozone trend isolated from the climate change impact. And then scenario 5 is encouraged. It's not required though for the modeling teams, but it's the same future scenario, but with 2015 CO2 so that we can see the ozone CO2 interactions. And using all of these scenarios, we should get a pretty good picture of ozone impacts under future climate change. And so just to familiarize people with the GGCMI protocol, we use harmonized crop management inputs. And so for water, we simulate both rain-fed and full irrigated conditions. This is based on the Mirka 2000 data set, and the irrigated fractions are adapted from Siebert et al., which is published in 2015, and these are held constant over time. For nutrients, we use mineral fertilizer applications derived from the Land Use Handbook 2 product for 2015. And we also use manure application inputs that are grid scale specific, but constant across the crops. And this fertilizer scheduling assumes that 20% is applied at sowing and then 80% is applied a quarter of the way through the growing season. And then we hold land use constant at the 2015 level to isolate both the climate and the ozone signal. And then we consider 2 tillage events on planting and harvest, 70% residues, no pests and diseases, no soil erosion, no multi-cropping, and no cover crops. So sorry, Willington, about no pest and disease. Hopefully we want to do that someday, but it's just the global data sets for that don't exist yet, like detailed ones for our inputs. But that's on the agenda. Ideally, we would like to add in all of these limitations. But for right now, this is the current GGCMI framework, so we adapted that for AgMIP also. And just an example of what the inputs look like. So this is a wheat mask. So we have these for not just the staple crops, but also for many more. So if you're interested in global gridded studies, I mean, please contact us or go to the GGCMI Redmine. You can find a lot of the maps and masks there. So on the left, we have winter wheat, and on the right, we have spring wheat. This is again from the Mirka 2000 cropland data. And in each of these grid cells is the irrigation fraction that I mentioned. So we know approximately how much of that grid cell is irrigated. And then on the bottom is in the example of the crop calendars, which was, these were created by Jonas Jägermeyer and the GGCMI team. So it's an aggregation of all the data sources. So on the right, you can see the data sources that they use, Mirka, Sage, and many others. And then it creates the typical planting and maturity dates for the different grid cells. And we have both for winter wheat, spring wheat, but again, we have these for many crops within GGCMI. And then last on the inputs is the climate models. So again, we have the three climate scenarios, the historical 126 and 370 scenarios. And then we're using two GCMs for these climate models. We're using the GFDL and the UK ESM climate models. Ideally, we would like to use more, but because hourly ozone is limited for CMIP6 data, we are only going to use two. And both of these model outputs are downscaled and bias corrected following the AgMIP framework, so the Intersectoral Impact Model Intercomparison Project framework that was developed by Stefan Lang. This was also done in GGCMI Phase 3. And the reason we do this is to get daily half-degree resolution global grid input. And it's bias corrected based on the ERA5 reanalysis dataset, which is a standard dataset that a lot of global modeling studies use for reference. So now to get into the results. So these are the decadal climate change and ozone trends. And so on the left, we have under the future SSP 126 scenario, and on the right is the future SSP 370 scenario. And so I'll start right now. So if you look, they're the same. The global average was 37 parts per billion, but then as it progresses through the decades, on the left, it's going to get much lighter because it decreases down to 27 parts per billion by end of century. And on the right, it got much darker because it increased to 41 parts per billion by the end of the century. And this is concerning because ozone stress can cause reduced photosynthesis and accelerate leaf senescence within the crops. And so having higher ozone concentrations is never ideal. And just a snapshot of one of the results. So this is from one model, a snapshot of the year 2050 for maize with an ozone-sensitive cultivar. We simulated the grid cells based on maize production that were, again, in that crop calendar that we have and the Mirka extent. And so then we compared the future ozone and climate change scenario with the historical ozone scenario, which is our baseline. I'm sorry, what was that?
One more minute.
Okay, got it. Yeah, I'm almost done. So preliminary results indicate that maize yields were projected to decrease by 19% by 2050 and by 41% by the end of the century. But again, this is just a snapshot, 1 year of one model. But what I do then is I aggregate it for all of the years. This is still just one model because we only have one model results so far, but we hope to have more soon. But so this is totally aggregated all the years for the SSP 126 scenario, which is the green bars, and the SSP 370 scenario, which are the yellow bars. And then the solid bars are for the ozone tolerant sensitivity, and the dash bars are the ozone sensitive classification. And you can see that there's some large yield losses due to both climate change and ozone impacts, which, I mean, these are, again, this is one model. It's a little higher estimates than what was observed from the GGCMI Phase 3 study, but we think with other models that will change. But again, like 20% losses in maize or 48% under SSP 370 is concerning. And then when we compare the future ozone with the 2015 ozone runs to isolate the ozone effect, this is when it gets interesting. We can see that there's a clear benefit to going for SSP 126, that there is a yield increase of 2% in maize and 5% in wheat. There's still a slight yield decrease under SSP 370, but part of that is because then when we look at the CO2 interaction, you see that this is the CO2 fertilization effect. It's much higher in wheat because it's a C3 crop compared to C4. But this effect that should be benefiting wheat is actually not because of the ozone impact. So there's not the impacts are not as pronounced. And with that being said, yeah, so just some conclusions to wrap up. The combined effects of ozone with high cultivar sensitivity and climate change can lead to large global productivity losses. The production of all four crops, I didn't show all four, but it's projected to decrease under the SSP 370 scenario. and the magnitude of losses depend on the cultivar ozone sensitivity. And hopefully by the end of this, we'll have some really cool global maps that can identify hotspots where regions are at risk for ozone stress. And we think ozone impact should be considered in future agricultural impact assessments. So with that, I'll end it, but it's also still ongoing. So if you are interested in joining, please let me know. We're still open for new teams. Thank you.
Thank you.
Thanks for a very short question. Someone has a question. So then we move to the next.
Okay, thank you.
Thank you.
Jose.
Okay.
Sim. Penso no ponto, não sei.
Okay. Hi everyone. My name is Jack Rawdon. I'm a researcher at UCL. Work with Marco Springman, and today I'm going to take you through something that's kind of maybe a little bit more not really been touched on this week yet, but hopefully you find it interesting. So, looking at pollination today and how understanding pollination dependence in wild plants can improve our understanding and hopefully move towards abilities to model feedbacks between kind of crop systems and these kind of natural systems. So first off.
Once you've done it the first time on the laptop, you'll figure out.
Yeah. So why is animal pollination important? So first off, it contributes to almost 90% of wild plants for their reproduction. It also contributes to 35% of global crop yield volume, and it's also very important for micronutrients. Vitamin A, C, and E are often derived from pollination-dependent crops. And I know in Southeast Asia, studies have shown that almost 50% of vitamin A intake come from very pollination-dependent crops. So it's very important. What is the global status of pollinators? Unfortunately, it's not looking good for them. There is kind of a lot of studies have come out which show these kind of global declines in bee species richness, and then also more kind of on a regional scale. We're seeing kind of declines in Britain, and there have been declines shown in the USA. So a lot of declines, and as a result, we need to kind of better understand this issue. So how can that decline in pollinators be linked to plant decline? So to understand and measure this, we come up with something called pollination dependence. It's a continuous scale between 0 and 1. So if a plant has a pollination dependence of, say, 0.8, that means that without pollinators, the seed set of the plant will be declined by 80%. And if a pollination dependence is 0.2, without pollinators, seed set declines by 20%. So all plants sit on this 0 to 1 scale, which means there are some plants which wouldn't be affected at all in a scenario of pollinator decline, and there are other plants that would be affected massively. And when a plant can't get enough pollination, pollen limitation occurs. And this is a widespread problem. Studies have shown that between kind of 30 and 60% of crop systems worldwide are pollen limited. So their yield has been compromised by not having enough pollination. And pollen limitation also occurred widely in wild plants. So we have these issues that kind of mirror each other in crop systems and natural systems. So we kind of need to build an integrated understanding of how these both kind of affect each other. And how do these co-declines impact people and nature? So firstly, pollinator decline can lead to direct yield declines for pollinator-dependent crops. Biodiversity loss, because if your reproduction in an ecosystem is compromised, that will cascade up through the trophic levels. There's also these indirect impacts onto crop systems. If you have a highly pollinator-dependent ecosystem, that will get degraded more rapidly when a pollinator decline scenario occurs. And if you have a degraded wild system, that will then kind of reflect onto a crop system. And if that crop system is pollinator dependent, then you have this kind of reflecting kind of decline in both sort of situations, which can obviously compound risk. And then obviously because pollinators prop up these ecosystems, you get a declining quality of ecosystem that will then have impacts on ecosystem services more widely. So that will affect kind of all these kind of things which we're familiar with in terms of ecosystem services. So what are we aiming to do here? So we generated a global map of pollination dependence in wild plants, which we refer to as naturally vegetated habitat here. We did this by taking an assumption in the literature that there's a kind of a phylogenetic driver of pollinator dependence. So if you have a plant that's got a specific pollinator dependent value, a closely related plant to that will also have a kind of a similar pollinator dependent value. So we took an empirical data set of 1,000 species of plant which have had their pollinator dependencies measured, and we used this phylogenetic assumption to model pollinator dependence for about 130,000 species of plant. We then took them and assigned them to species distribution maps and stacked them together to create this kind of average pollinator dependence in wild plants for kind of every grid cell on Earth. So we use this data set to answer a few questions. So firstly, how does pollinator dependence vary across the Earth's surface? So we have the data for cropland systems often for pollinator dependence. So by combining cropland pollinator dependence and wild pollinator dependence, you can build this kind of integrated understanding of pollinator dependence across the Earth's surface. And then with this knowledge, kind of how does this then affect biodiversity, crop yield, and wider human well-being? So this is kind of the big map. So by combining crop pollinator dependence and kind of wild plant pollinator dependence, we've come up with this kind of map of the Earth and kind of pollinator dependence distributed across it. So kind of the big result is this latitudinal gradient. That's driven by wild plant pollinator dependence. It kind of transpires, and this is reflected in literature, that the closer you are to the equator, the more pollinator dependent your ecosystem is. So you can kind of see that highlighted here. But you can also see where these kind of arable crop systems are dominant. So in North America and in Europe, where a lot of the agriculture is wheat or maize, these crops aren't pollinator dependent at all. So this is reflected in this map. And then, of course, where you have ice sheets and you have deserts, there's no pollinator dependence because there's no vegetation there. So that was interesting to see, and it kind of firmed up that pollinator dependence is massively varied a lot where depending on where you look on the planet. So looking next to biodiversity loss, we compared our pollinator dependence of wild areas to species richness in these wild areas and the plant diversity in these wild systems. We found that wild plant species richness is closely coupled to pollinator dependence, which is really interesting. And it kind of shows that in these really kind of these tropical areas where you have a high biodiversity, your pollinator dependence is also really high. However, that isn't completely kind of a one-to-one relationship. We kind of compared the 90th percentile of the most biodiverse areas and the most pollinator dependent areas, and there are some differences. But they do tend to overlap in kind of Indonesia and in large stretches of kind of the Amazon. We also kind of summarized it at the biome level, and you can see these 4 pink boxes right at the top here are the most pollinator dependent biomes, which are tropical, subtropical forests. So essentially the tropics, very vulnerable in the kind of the wild systems. So bringing that across to crop yields, what does that tell us? So we did a similar thing by kind of overlaying where are the most pollinator dependent crops and where's the most pollinator dependent nature and where does those kind of areas overlap? Once again, kind of Indonesia is really kind of is showing up quite a lot. So in Indonesia, by the looks of things, in agriculture, pollinator dependence tends to be high. That's driven quite a lot by oil palm, which is a pollinator dependent crop. And the nature around it is also pollinator dependent. So, they are kind of Indonesia is flagging up as quite a risky place to pollinator decline because you might see these compounded risks where both kind of pollinators are affecting ecosystems and the natural systems that are and the crop systems that are adjacent to them. There is less of a correlation between kind of crop PD and kind of wild pollinator dependence, but there is kind of a small kind of positive correlation there. And then kind of looking more kind of granular at the different crops that are most affected, oil palm comes out on top. So over 3/4 of global oil palm kind of crop is surrounded by the most pollinator dependent natural systems. So that's a potential risk. Vanilla also shows up, but vanilla is less coupled to the environment because often it's hand pollinated. But crops like nutmeg and cocoa and cashew, which are all very pollinator dependent, also show up here. So yeah, reason to be concerned. And then finally, to quickly look at human well-being, we derived an index that measures how close are people living to nature and how pollinator dependent is that nature. So at a value of 1, you have people living the closest to nature, and that nature is the most pollinator dependent, and vice versa. We then overlay that onto a kind of a gridded population density data set to understand kind of where is the highest risk, and kind of where are the highest kind of how much the population is affected. So you can again see that the tropics, and particularly the African continent, has this kind of the highest risk of kind of to pollinator decline. And you can see that a large amount of the global population is probably affected by a pollinator decline scenario. So kind of what are the key takeaways from this? Well, pollinator dependence vary dramatically across the Earth's surface. Depending on where you're looking at on a kind of a more local scale, you're kind of going to have a very different conversation. Ecosystems with very high species richness also have a high pollinator dependence. Key pollinator crop dependent crops may be further threatened by pollinator dependence in adjacent ecosystems, and the majority of people live very close to pollinator dependent nature. So from the crop yield perspective, this is very much like a very kind of early effort at this. It hasn't really been quantified before, but I hope this is the first step into trying to make some kind of modeling framework that will allow for the kind of these interactions between crop systems and natural systems and both of their respective pollinator dependencies to be more kind of accurately kind of quantified, so we can get a better sense of what risk this might pose to us. So yeah, thank you for listening. My contact details, please, if you have any questions.
First off, thank you so much for this. It's so nice to see these sort of ecological modeling approaches at AgMIP, and I think we need more of that. And so thank you for making that bridge and coming here. I guess I have just a basic conceptual question. So you're linking pollination dependence in ag lands to their adjacent ecosystems and saying if they both have high dependence, then there's higher risk.
Yes.
I think there's one way to look at it potentially, if that is a little bit different, but I'm interested in what your thoughts are, which is if there is ag land that's pollinator dependence that is next to areas that have no pollinator dependency, that's potentially a very high risk because there's none of that supporting pollination activity adjacent to ag lands. Like, is there not a resilience baked into having pollinators in ecosystems next to ag lands because they're dependent, if that makes sense?
Yeah, it does. It's a really interesting question. I feel like, I guess, pollinator dependence in ecosystems may not necessarily be tied to pollinator diversity and abundance in those ecosystems. So you might have an ecosystem that isn't actually dependent on pollinators, but that won't stop loads of invertebrates and bees or animals from living there that can then pollinate crops. We're still kind of trying to puzzle kind of the evolution of pollinator dependence in ecosystems and what kind of that has on kind of animal communities that live there, kind of how those are linked. It's all kind of quite new science and it's all kind of in its infancy, but it's a really good point. I think it'd be interesting once we kind of get more of a handle on how like the existence of pollinators in an ecosystem and whether plants have kind of co-evolved to support that, whether that can then have an impact on crop yield. That's really interesting.
The relationship between the dependence and the presence of.
Yeah, exactly. Yeah.
Thank you so much for the very interesting presentation. I have many questions. I will try to do two short ones.
Yeah.
The first one is we have trends globally around increasing size of fields and around the bigger fields and the land use expansion. So the first question would be, are you planning to do some link to land use change of somehow? That will be the first one. And the second one is in your map for the agricultural land. I was a little bit surprised to see the European Mediterranean, Western Argentina, California, where you have a lot of areas with fruit trees and nut trees that are corn dependent not showing up. So I was wondering, is it an artifact of the list of crops you have, or do you have any ideas?
Yeah, two great questions. First off, I think this bit of work is kind of leading into some wider stuff we're doing in the team. We're trying to, I guess, conceptually kind of look at ecosystem services and try and check, like maybe adjust how we look at them. I think a lot of the time when you do these ecosystem service models, you use natural habitat as like a proxy for different services that might exist. And often, I mean, in Invest for pollination service, there is kind of local scale kind of grading of natural habitat based on certain criteria and stuff like that, but there isn't kind of a more general kind of mechanistic way of kind of seeing, kind of digging into that natural habitat and saying kind of what are the qualities of the natural habitat which might aid or abet the ecosystem service. So I think we wanted to give more context to nature and its dependence on pollinators, and that's where this kind of came from. But yeah, this is also leading into trying to then understand, you know, how is this more locally going to be linked to land use change, and how can we start to kind of build that into things. And your second question about those regions, yeah, absolutely. And they are, if you look at the full pollinator dependent crop map that's continuous, they absolutely are highly represented. It's just that in the 90th percentile, yeah, these tropical crops, which are like almost 100% dependent on pollinators. So they were the ones that kind of got flagged up at the very highest echelons of pollinator dependence.
We can catch up later.
Yes,
I'm really happy to chat about it next.
Henrik Haas. Yeah. Okay. You wanna share your screen?
Sure. Should be coming up. Hope you guys can see it.
Yeah, it's
Cool.
here.
Nice. Let me get my pointer in here. All right. Yeah, my name is Enrique. I am a postdoc at Columbia University, and I guess my presentation might be a little bit different because I'm not a crop modeler. I'm not an agronomist. I'm actually a hydrologist, but I work closely with the AgMIP team in New York. We are collaborating on a watershed in upstate New York, and today we're going to share a little bit of our preliminary results and how we are kind of bridging the gaps between hydrological modeling and ecosystem modeling to better capture water quality dynamics. So just a little bit of background on our project. We are working in the New York City watershed system. It is a beautiful piece of engineering that comprises almost 19 reservoirs that transport water from all the way in upstate New York to almost 200 miles downstream to the city, and supply high quality drinking water for almost 9 million people. And 90% of this delivery is actually done without any kind of filtration, which saves the city millions and millions of dollars. Now in this project, what we are mainly addressing, it's the potential challenges posed by climate change on water quality and how much climate variability might impact agriculture best management practices and also agricultural productivity across the watershed system. The water quality
It's frozen here as well.
What?
Well, this is the nightmare.
So maybe I can ask you a question.
Yeah, please.
You said so easily in the European and the American North American areas, the pollinator dependent crops don't play a central role. I do not quite agree.
I mean, I think
in some crops that need pollinators.
Yeah, no, sorry. The kind of in terms of how dependent on countries, I think they are very dependent in the measurements, and to verify like this California almond which obviously has a huge amount of hives just to deal with it. It was more about kind of, I guess, we took 90th percentile of pollinate dependence, like average pollinate dependence in croplands in the world. And in tropical areas, that's where it just was highest because
I can see that. That sounded a bit different than something that is last year. And the spatial extent of the ecosystems, that not also matter. The natural.
So we, in order to make that more map, I went down to 10-meter resolution and basically classed, land class into either
natural land, cropland, or non-vegetated land. And then I worked out what fraction in each 20 by 20 kilometers had those two classes in, and then I weighted this out by the pollen dependence value associated with that area. To make a constant map.
But you didn't account for, maybe I don't get it, but didn't account for the size of the ecosystem, the English spatial extent of the ecosystems in calculating that. The possible links between the two.
Yeah, I mean, there's definitely like ways we can kind of continue with this. Like, I think it's just very much a first it's just happening. But yeah, no, I'm really excited.
Yeah, we have that in or mix up the land exchange maybe natural natural ecosystems in that area problem depending how big you make it. Look at it.
I mean, this is the classic. So there's the Lonsdorff kind of function. It's like the further away you get from the natural, the less the kind of your being pollinators. This is kind of, I guess, this was about the kind of the intrinsic pollinator dependence that the plants there, rather than kind of how much sufficiently pollinated they were going to be. But pairing this up with something that was like pollinator sufficiency, or to get it. And then, you know, the other side, the next side is the pollinators themselves. And we're looking at it from what's kind of the baseline pollinator dependence of the plants, but we have this massive gap now, which is where are the pollinators, what they're actually doing to those plants, and their pollen limitation current out. So yeah, there's so much that
and certainly if you want to do that globally.
Yeah, exactly. And the other thing with pollination is it is such a local thing. I think there are these macro patterns in pollination. I mean, it is justified to look at it at a global level, but I think ideally you complement that. So you'd have these big global patterns where you're saying, hey, move towards the equator, pollinator dependency of ecosystems goes up. But within that, there's a lot more subtlety when you zoom in, and combining that happens.
Sorry guys, they had a problem with his internet. So here we have internet that apparently had a problem with his connection. So you're just waiting 2 more minutes if he connect again. Otherwise, we send a message to him to record his presentation and send later.
Yeah.